I also want to look at another example of mutations. This may make it a little bit easier to understand. This is not a sequence of mRNA, but rather this is just a sentence. And I want you to understand how a point mutation and frameshift mutation may work in a sentence, which may help you understand a little bit about how they would work in an mRNA or DNA sequence. So remember, a point mutation is one where just one nucleotide has been changed. So we're just going to change one letter for this. So let's just change this A to an I and see if the sentence still kind of makes sense. So instead of the fat cat ate the rat, it is now the fit cat ate the rat. Most of the words are still the same, and in fact the sentence pretty much still makes sense. Why don't we do a frameshift mutation where we insert the I into it rather than just changing the A to an I? So this is going to make things a little bit different. Remember, frameshift, we're going to shift everything over. All right, do you see how this works? Remember, they're broken down into 3 letters because that's like codons, right? Codons are 3 nucleotide sequences. So the fiat-cat-eth-erat. All right, do you see how the frameshift mutation makes a much larger difference in the comprehension of the sentence? Same way with amino acid sequences. All right, it works the exact same way. And that's what we're going to look at here in just a few seconds. Hi. You know, I was sitting here thinking about what exactly makes Angry Birds so angry, and I don't think it has anything psychological, but rather it's probably some type of maybe mutation that they have that just makes them naturally angry. I mean, have you ever seen a happy bird? No. Probably wouldn't sell a lot anyways. You have Angry Birds, and what makes Angry Birds angry is genetic mutations, and that's something I want to take a look at today. Because genetic mutations is important to understand because they can cause a whole host of problems inside the body, including genetic disorders and possibly cancer if they're coded in the right region. So, what I want to do is to get a little bit of understanding of what different types of mutations there are, how do we recognize them, and how do we tell if they actually make a difference in the proteins that are being produced. So let's take a look at this example here. You notice that I've already cut them into 3 nucleotide sequences. If you remember, those are called codons, right? So you've got AUG, CCA, UCC, AGG, UAA. Well, let's do what's called a point mutation first. And a point mutation is when one of the nitrogen bases is altered or changed to a different nitrogen base. And this could be for a whole host of reasons. But let's just say that this right here gets changed to something different. Let's say instead of UCC, it ends up being ACC. All right, so we're going to write this out. And the point mutation is here. So instead of a U, we're going to have an A. And then the rest of it is the same. So when we go and evaluate this, we need to look at our amino acid chart to be able to determine what amino acids are present here and to see if it would actually make a difference, because some point mutations don't actually change the amino acid sequence. So let's take a look here. We know that AUG is our start codon, and that's the same. All right. If we look at our little chart, we'll see that CCA is proline, and that doesn't change. Here's where we see that there's a difference. So UCC, we're going to see that is serine. And again, all these are just different types of amino acids. Those amino acids build to make the proteins that are necessary for your life. So let's look here. We got ACC. ACC is threonine. All right, notice that there is a difference here. This is not the same as this. So the amino acid sequence is different. The protein is going to be different. All right, so let's look here. We've also got AGG. Let's go ahead and finish this out. And AGG is arginine, which is the same. And then UAA is your stop codon, which will tell the amino acid, or will tell the sequence to stop reading. All right? So we look here, there is a difference here. This codes for threonine, this codes for serine. So this is a mutated RNA sequence, and this sequence will, as a result, not code for the correct protein that is necessary for life. All right? So that is an example of one point mutation. You'll notice in this example here, we have a second point mutation, but we've moved it to a different spot to illustrate a different point. Not all point mutations cause a change in the amino acid sequence. You'll see what I mean here in just a minute. So notice that now this C has been changed to a G. All right, this is a different RNA sequence here. So let's go through, code it again, and see if there's any difference between UCC and UCG. See if that causes anything. We all know that this is going to be start, and that this is proline. All right, but we look at UCC and UCG. UCC is serine. If we look at UCG, UCG is also serine. Okay, UCG is also serine. What you'll notice is that on that amino acid chart, there are several 3-nucleotide sequences for each amino acid. All right, so sometimes there may be 4 different 3-nucleotide sequences that code for the same amino acid. So if they change in a way where it doesn't cause the amino acid to change, that's actually not necessarily a good mutation, but it's a mutation that's not going to cause a problem in the future. So that's certainly something that's important to understand. All right, so you can have a point mutation where it does not change the amino acid sequence. Talk about being lucky, right? The other type of mutation I want to talk about is what's called a Frameshift mutation. And this is classified as either an insertion or a deletion, whereas a nucleotide is either inserted into it and pushes everything to the right, or one is deleted and pushes everything to the left. You'll see what I mean here in just a minute. So let's say, for example, that we have a Frameshift mutation here, all right, where the circle is. And we're going to insert a new letter, all right? So think about what that's going to do. So we start off with the same sequence as before, but instead of UCC, let's just say we're going to add an A. So what that does is that pushes all of the 3 nucleotide sequences over one, all right? So if we insert something here, this C is going to be the first part of this now. So it's going to be CAG, and then GUA, and we're going to have an extra A at the end. You see what I mean by Frameshift? It shifts all those nucleotides over one spot. And think about what may happen with this. Think about the impact that this might have on coding for the protein. UAG is still your start codon, and CCA is still proline. But now we have UCA. UCA is still serine, luckily enough, but instead of AGG, we have CAG. And CAG is glutamine. And instead of a stop codon, we now have GUA. GUA is valine. And then we have this extra letter here. So you see what happens when we have a Frameshift mutation. Frameshift mutations cause every single amino acid after that mutation to be different. All right, if this continued for 100 amino acids, odds are most of those are going to be different. Now that is a significant impact compared to a point mutation where only one amino acid is altered. All right, a Frameshift mutation is going to alter every single amino acid after that mutation. So obviously Frameshift mutations are much more dangerous and can cause much more problems than just a simple point mutation. All right, that point mutation may even be in a non-coding region, whereas this, you can alter start codons, you can alter stop codons, you can alter every single codon after that mutation. All right, so just some things to think about as you go through this. Okay? If you have any questions, make sure you let me know.